Good morning. Myself Ayushi from group 319. My today's topic is the regulation of the genes. The genome of the eukaryote are very complex. They contain repetitious genes and inverted genes, palindromes. The genetically function portion of the DNA alternative with non-functional portions, that is spacers. There are transcribable and monotraceable species. It has been recently found that even within the gene, the functional portion of the DNA, there are intervening non-functional portion called the introns in this case. Georgiev and his co-workers have found that eukaryotes have been assembled and found that the eukaryotes have been assembled the whole regulatory system transcription. The transcription gives rise to the pro-mRNA, only a small part of which is a true mRNA responsible for protein synthesis. The greater part of the pro-mRNA is non-informational and degrades under the action of the nucleases. The breakdown of the part of the pro-mRNA and the separation of the two mRNA shown schematically in figure 8.14 and are called processing. Thus, the transcription consists of a very large genetically non-functional zone of the DNA, which may be called the acceptor gene, and the smaller structural zone. It is exactly the acceptor zone that contains portions, that is a promoter region, to which RNA polymerase attaches itself. The movement of the polymerase along the transcription length translational is regulated by the interaction of the regulatory proteins with acceptor regions. The transcription model provides a much subtler regulation of gene expression, blocking and activation than the regulation of the genes. And in prokaryotes, the interaction of NHC protein and histones with acceptor regions affect the structure of the chromatin and chromosomes, determining the packing of DNA, on which the possibilities of the transcription depend. A single transcription contains various acceptor regions which recognize different protein, and conversely, different transcription can recognize some protein. Accordingly, the addition or the separation of one protein can turn or off the entire assembly of transcriptions. The large size of the DNA acceptor zone turned out to be necessary precisely for a fine and diverse regulation, without which no differentiated multicellular organism would be exist. According to the transcription model, mRNA is at the end of the giant pro-mRNA, since there are acceptor regions which are identical for different transcriptions. The initial part of the pro-mRNA must contain identical sequence for different pro-mRNA. This has been confirmed by the experiment. Origel and Crick 1980 formulated the hypothesis of selfish DNA. The greater part of the DNA in the cell of the higher organism is not functional. It has no effect on the phenotype. It might be expected that the selfish DNA is replicated independently, behaving in this sense like a not too harmful parasite. The presence of an excess such DNA can, however, slow down the state of meiosis, and then the size of the genome set up a limit beyond which the development cannot be speeded up. The problem of the regulation of the gene expression in eukaryotes have only recently become the concern of the biophysics. The complex nucleoprotein system of the chromatin and chromosomes have not been studied sufficiently well for a clear-cut formulation of many physical problems to be made possible. At the same time, the physics made it possible. At the same time, physics mathematical modeling of regulatory process of the development is under processing. Here we can see in this diagram. It is the representation of the process transcription. Is the piece the promoter gene. A1 to AM are the acceptor sites. SM are the structural genes. It is the promoter. A1, A2, A3 and till AM they are the acceptor sites, and SM they are the structural genes. Nucleases, it converts into the non-informative zone. The nucleases, it's the pro-mRNA. Pro-mRNA is converted into the a single mRNA. How it is converted? With the help of the enzyme nucleases. Transcription is the process in which after the replication of the DNA, the protein is transcripted. It is in the form of the zones, like acceptor zones, structural zones. Acceptor zones, they accept the protein that coming through, and the structural zones, they and when it comes to structural zones, it codes for the structures, like SM, it codes for another structure, and they just codes for the another structures. And also here it also comes the informative zone, which give non-informative zone. Non-informative zones, here it is, they don't get any information about what the being is happening or what going through. This is the systematic or schematic representation of the process transcription, which contains promoter genes, acceptor sites. Acceptor sites also promoter that promotes. There are also factors in the process of the transcription. That is initiation factor, elongation factor, and the factor which also end this process. This is the systematic representation of the transcription. Thank you so much.